Hello. In this video we will introduce you to a new technique that allows for sensitive detection of cell surface antigens, the clamp method. Each of these topics will be covered in this video. However, let's begin by reviewing the conventional method so that we can compare against our new method. Antigen detection via antibody binding is a widely used method to differentiate and analyze various types of cells within our bodies, allowing us to distinguish between cell types and detect abnormal cells in a sample. For example, lymphocytes in the blood include helper T cells and killer T cells. There is no difference in their internal structure or size when observed under a microscope. However, the cells can be stained with fluorescently labeled antibodies that bind to either CD4 or CD8 antigens, which are specifically expressed on the surface of each respective cell type. With this method, we can fluorescently distinguish cell types based on cell surface proteins exclusive to the cell types. As the diagram illustrates, the conventional method, using fluorescence-labeled antibodies, involves indirectly binding fluorochromes to the cell surface antigens of interest. We will now explore the limitations of this design and discover why this technique is difficult to use for surface antigens with low expression levels. The images on the left illustrate the biggest weakness of the fluorescence-labeled antibody method, specifically that the intensity of the fluorescence produced is dependent on the amount of antigen present on the cell surface. If the antigen is highly expressed, as we see in the rightmost fluorescence image, then it can be clearly observed. However, if the surface antigen has low expression, it can be difficult to determine whether the antigen is present or not. The figure on the right illustrates the concept of cancer immunotherapy, in which antigen recognition is very important. The target antigens for the therapy are usually ones overexpressed by cancer cells, because recognizing them is easy. Meanwhile, studies in companion diagnostics aim to determine the exact expression levels of specific antigens so that we can predict drug effectiveness. This practice is more reliable with more sensitive detection, and it is for these reasons that we've developed technology to improve the sensitivity of our antigen detection methods. And now, let's have an overview of the clamp method. The purpose of the clamp method is to enable highly selective and highly sensitive fluorescent labeling of cells with specific cell surface antigens. This technique involves a primary antibody designed for a particular cell surface protein, a beta-galactosidase labeled secondary antibody, and Mugf, a fluorescent substrate for beta-galactosidase. This fluorescent substrate, Mugf, is actually non-fluorescent and non-permeable to the cell membrane in its beginning state. However, if the primary antibody binds to an antigen-presenting cell, the conjugated beta-galactosidase will convert Mugf near the cell surface into a compound with a quinone methide structure. This compound is then permeable to the cell membrane. It will enter the cell and react with thiol and aminol groups inside, forming covalent bonds and emitting fluorescence. Large amounts of Mugf are converted and subsequently accumulate in the cell, creating an intense signal. In this way, the mechanism is very highly sensitive and will produce strong fluorescence even with very low levels of antigen expression. Now, we can discuss experimental operation and what the clamp method can accomplish. The typical clamp method experimental procedure involves treating cells with the primary antibody for 1 hour, and then with the beta-galactosidase conjugated secondary antibody for 1 hour. The cells are then treated with Mugf, a beta-galactosidase substrate, for 30 minutes before being washed and measured for fluorescence. The clamp method can be applied to a variety of samples. For live cells, only surface antigens can be detected because the antibodies cannot enter the cells. For PFA-fixed cells and tissue sections, however, intracellular antigens can be stained as well. Additionally, the stained samples can be stored. For living cells, PFA fixation is required after staining, but we've confirmed that the samples can be refrigerated for 1 week. This is an advantage when it is difficult to perform fluorescence imaging or flow cytometry immediately after staining. First, let's demonstrate clamp's compatibility with live cell samples. In this example, our sample cells are live A549 cells, and fluorescent staining is being used to detect CD44, a specific cell surface antigen. Among the staining images here, we have clamp method staining on the left, conventional fluorescence labeled antibody staining in the middle, and an image with no stain on the right, our control. As you can see, the clamp method clearly produces fluorescence, while the conventional stain showed almost no fluorescence. This is evidence of the higher sensitivity that the clamp method possesses. To the right of those images is a bar graph quantifying the fluorescence intensity from the images on the left. This reveals the magnitude of the difference in sensitivity between clamp and the conventional method. Next, let's look at the fluorescent stained images themselves. In the upper left, we have a fluorescence image of a cell stained by the conventional method. We can see how the target antigens are scattered on the cell surface. Meanwhile, in the lower left, we have the image produced by clamp method staining, which shows uniform staining of the entire cell. This is because the dye, after being formed by beta-galactosidase-mediated Mugf reaction, enters the cell and binds to intracellular proteins. Because of this, the clamp does not provide information on the localization of antigens. The clamp method is more appropriate for experiments such as flow cytometry that intend to measure the amount of antigen expressed, or for experiments that intend to confirm the presence of antigen, even in small amounts. Here we have another example, now using live HepG2 cells. The surface antigen we are looking for is PDL1, a known cell surface marker for cancer. HepG2 cells do not normally express PDL1 on their surface, but are known to do so in the case of certain cancers. Alternatively, INF-gamma can be used to induce cell surface expression of PDL1. INF-gamma treatment was used, and we performed an experiment to verify that only cells with induced antigen expression were going to be stained. Here we have the results of the experiment. The top row of images are the fluorescent results from each combination of staining method and treatment condition. The two images on the top left are ones produced by the clamp method of staining, where we can see that fluorescence was observed only after INF-gamma treatment. The two images on the top right were produced via staining with a fluorescently labeled antibody. Here, we can detect hardly any fluorescence regardless of INF-gamma treatment. To the right of these images, we have quantified measurements of fluorescence intensity from the images on the left. We can see that fluorescence intensity did increase with the conventional method, but if we compare the effects of INF-gamma treatment between the two methods, it's clear to see that the clamp method is far more sensitive. Following the last experiment, we decided to test whether we could specifically stain only antigen-expressing cells even when mixed with antigen-negative cells. In order to correctly identify our antigen-negative cells, we labeled the non-INF-gamma treatment HepG2 cells with the green fluorescent dye CFSE. These cells were then co-cultured with INF-gamma treated cells and stained via the clamp method, blue. Here we have the results of that test. The left fluorescence image illustrates which cells express PDL1 on their surfaces and have been stained by clamp as consequence. The middle image shows us which cells have been stained by CFSE. On the right we have the merged image. As you can see, the stains are mutually exclusive in each cell, which shows that clamp has selectively stained only the PDL1 expressing cells. Next, we will present the results of clamp staining upon PFA-fixed cells and frozen tissue sections. Previously, we performed an experiment by staining live cells that express the CD44 cell surface antigen. Here, we repeat that staining process, except our CD44 expressing HeLa cell samples are either live or PFA fixed. With these results on the right, we confirm that the clamp method can be applied to fixed cells as well. Now, we should see how the clamp method works with FFPE, formal and fixed paraffin embedded tissue sections. In this example, we have FFPE sections of the human small intestine. Clamp has been used to stain muscle fibers in the tissue, blue, and TSA, another sensitive surface antigen detection method, has been used to stain keratin, red. Together, the results clearly illustrate the myofibers in the tissue. They also prove that clamp can be applied to FFPE tissue sections, and that clamp can be used to multiplex with other methods like TSA. So far, the data presented has been from fluorescence imaging, but the benefits of clamp extend to flow cytometry, improving that method's sensitivity as well. The structure of the clamp dye, Mugf, is shown here. In the graph to the right, Mugf's excitation spectrum is represented by the blue line. As the line indicates, it is difficult to excite at 405 nanometers, which is very often used as the exciting wavelength in flow cytometers. Because of this, we at Dojindo developed a new Mugf molecule that is more suitable for 405 nanometer excitation, represented by the orange line in the graph. We named it Mugf3. Now let's see the results of flow cytometry using Mugf3. In this new experiment, we are once again staining PFA-fixed HeLa cells for CD44 antigen expression via the conventional and clamp methods. However, this time we are also performing the clamp method with Mugf3. In the graph, we see the conventional antibody results displayed as yellow peaks, the Mugf clamp results as red peaks, and the Mugf3 clamp results as green peaks. As can be seen, Mugf clamp, despite being unsuited for flow cytometry, still produces fluorescence intensity about 10 times higher than the conventional method. This reflects its very high sensitivity. Mugf3, which was actually designed for FCM measurement, is 10 times even more sensitive than Mugf. We also have fluorescence images on the right showing that Mugf3 is not only highly sensitive when used for flow cytometry, but also highly sensitive when used for fluorescence imaging. In order to emphasize Mugf3's usefulness, we have another experiment here that aims to replicate a situation useful for cancer immunology. Again, we aim to detect a low expression antigen. In this case, our sample cells are PFA-fixed MOLT4 cells, and this antigen of interest is the PD-1 antigen. PD-1 is an exhaustion marker in immune cells, and PD-1 measurements are important for understanding the efficacy of immune cells. The results of this experiment reveal a very slight shift with the conventional method and a much stronger shift with the Mugf3 clamp method. In these fluorescence imaging results, we see that fluorescence is too insignificant to be confirmed with the conventional method, whereas we can see significantly intense fluorescence produced by the Mugf3 clamp method. This experiment illustrates that antigens expressed in concentrations too low to be detected by the conventional method may be detected by the Mugf3 clamp method. Now, to summarize, we have developed a new, highly sensitive fluorescence staining method that targets cell surface proteins. Using this technique, we found that we could stain cells for specific cell surface proteins with higher sensitivity than the existing methods involving fluorescently labeled antibodies. In addition to our sensitive dye Mugf, we have also developed another sensitive dye, Mugf3, for use with flow cytometry. In the future, we plan to acquire data by using clinical samples. We also plan to develop green and red fluorescent dyes for the clamp system to complement the blue one seen here. As well, we hope to develop fluorescent substrates for enzymes other than beta-galactosidase. There are published references for this technology, and you can contact us for additional information. This is the end of our presentation on the novel clamp method technology, although there is more we would have liked to share. If you are interested or have any questions, feel free to visit our website or contact us. Thank you very much for your attention.